In this video, we're going to take a look at more titration curves. In the last video, we explained about buffers and why buffers resist change to pH. The buffers are made out of solutions of a weak acid and its conjugate weak base. If you're making that solution, it's kind of best to be able to mix it with the same concentration of both the weak acid and conjugate weak base. And now we're going to take a look at some more titration curves and make sure that we've got the components and parts of them identified. Now this is a titration of sodium hydroxide titrated with hydrochloric acid. So the with this means that that is the titrant. That's the stuff in the burette. And you can see that we end with a pH that's quite low. Now this pH curve is highlighting that we would select an indicator because it sandwiches essentially the equivalence point. So I want an indicator that's changing color on either side of the equivalence point. And the equivalence point is kind of like halfway up the steep part of the curve. And so here it's a pH of about 7. Acid-base indicators are listed in your data booklet. And you can see that at a pH of 7, bromothymol blue changes at 6.0 to 7.6, so that would be a good indicator. Or maybe even phenol red would work because it's 6.6 to 8.0. And essentially what I want is I want that equivalence point to sit in the middle of those two numbers. So it changes from one color to another color, right? And so if I'm adding base to a solution here, if I'm adding base to a solution that has bromothymol blue, it would start off in the acidic form, and as base is added, it turns blue. And you would stop the titration when it's green, because that's a mixture of both yellow and blue, so that's in between these two numbers. If we were doing that with phenol red, and so we had some acid sample that we were titrating in some base, it would start out as yellow and it would go to red, and I'd want to stop it when it's orange, which is a blend of those two colors. Now when we look at the titration, or at the indicator table, they've got these sort of fake formulas. This is the acidic form of bromothymol blue, and this is the basic form. The acidic form happens at 6.0, and at 7.6 you get the basic form. And the colors correspond. This is the acidic form, and this is the basic form. And finally, they give you a Ka for the indicators, which could be used in a question as well, because we've now dealt with Ka to find hydronium ion concentration. And so the indicator table has a lot of useful information. If I had, say, a titration that had an equivalence point at 9.0, phenolphthalein would be an appropriate indicator for that, because it fits in between them, right? In between 8.2 and 10.0, and I would expect that it's a blend of pink and colorless, so it'd be very faint pink. So here they picked bromothymol blue because it changes on either side of that, and this particular titration has two flat parts, but neither of those are actually going to be buffer regions. And the reason is that up at the top here, it has strong base, right? And at the bottom, it has strong acid. So there's no portion of this where you've got a weak acid and its conjugate weak base that has a flat part of the curve. So neither of those are actually considered buffer regions. This is our equivalence point, and if we were doing this titration in the lab, we would stop it at this point because it would produce a green color. Now, this is a little summary of acid-base indicators, as I'd mentioned. So phenolphthalein is an indicator. This is the acidic form. This is the basic form. It's an acidic form at 8.2 and less. It's in the basic form at 10.0 and higher. In between, it's a mixture of the two. In the acidic form, it's colorless. In the basic form, it's pink. And so we would expect that if it was in between those two numbers, so if it was like a 9.0, it's going to be a mix of colorless and pink, which is just light pink. Now, polyprotic entities are a little bit tricky to explain. Polyprotic acids, like sulfuric acid or phosphoric acid, or carbonic acid. So carbonic acid is H2CO3. It's a polyprotic because it has two hydrogens to react with water. So it could make a hydronium and a hydrogen carbonate. And then the hydrogen carbonate could also react with water to make another hydronium and carbonate ion. Now, this is a much stronger acid than this, so I would expect that this is going to go more forward, still less than 50%, but this will be much less than 50% because it's a much weaker acid. But there's still two hydrogens that can be donated, so it's called a polyprotic acid. A polyprotic base, on the other hand, would be something like maybe SO3 2 negative. So we could get that from sodium sulfite. And the sulfite ion, when it reacts with water, will make a hydroxide ion and a hydrogen sulfite. And then the hydrogen sulfite can react with water to make another hydroxide and a sulfurous acid molecule. And so because we can make two hydroxide from it, the sulfite ion is a polyprotic base. It can accept two different hydrogens. Now, in both of these cases, because it was a polyprotic, this chemical, hydrogen carbonate, could both gain a hydrogen ion, so it could act as a base, it could accept it, or it can donate this one, and so it could act as either an acid or a base, and so we call that an amphiprotic species. Same with hydrogen sulfite here, right? It could both donate or accept the hydrogen. Now, this curve is a curve that has sodium carbonate, and that's a polyprotic base with a strong acid. So I'm ending it with a low pH. Down here, this will not be a buffer, and the reason is that at this point, basically you've got hydronium ions and carbonic acids, but up here, this is a buffer region and this is a buffer region. They've flattened out the curve, and it has this weak base and its conjugate weak acid. Over here, it's got this weak base and its conjugate weak acid. And so those two are buffering regions. We also have two equivalence points. Now here, they've only measured one of them using methyl orange, but there are two places where the base and the acid have equal concentration, right? That's here. And then here, they've got another case where they've got base and acid are in equal concentration. So there are two equivalence points and there are two buffer regions. Frequently, these curves have a little weird tail. So this little weird tail I'd like to just quickly explain. Now, carbonate and water will make a little bit of hydroxide. So the initial solution that you start with of carbonate ion is basic, and there are some of these hydroxides. So those hydroxides will quantitatively react just right here with the hydroniums from the acid particle to make a water. And so we get this, or two waters. And so we get that happening here, but once these are used up, then we start getting this reaction where we get the carbonates and the acid particles doing this. That's what's occurring in this section over here. I guess probably more accurate in this section here. Okay, so polyprotic acids tend to have more than one equivalence point. they tend to have more than one buffer region. Almost all of these curves that we look at are going to have the tail end, because we're using either a strong acid or a strong base. Those are typically not buffering regions, even if they're flat. So here's the breakdown of phosphoric acid, which is another this is a polyprotic acid, and this time they're adding strong base to it. And we get that weird little tail. We get a buffer region, a buffer region. This is not a buffer region because it has hydroxides. We get an equivalence point and an equivalence point. Now, the phosphoric acid only has two obvious equivalence points, and it only has two obvious buffer regions, but it still looks like it has three hydrogens that can react. And the reason that we don't see the third equivalence point is a little bit subtle, and it's not super important that you get this if you've got all the other stuff. But what's kind of important to know is that the phosphate ion and the hydrogen phosphate ion and the phosphorus acid or H diphosphate, where are you now, have some, these two are so similar in pH. They're both essentially significantly basic enough that we don't actually see them as a separate bump on the curve. And so we're only seeing two of the reactions because two of the states, like the HPO4, H2PO4, the HPO4 2 negative and the PO4 3 negative, are both so similar in basic quality that they actually look like they're one like sort of stretched out part in the curve. So want you to be able to go through and look at some of these curves and make sense out of them. One thing that I'll draw your attention to is this. It's a little bit baffling to look at, but I want to just go over it quickly. If I had a strong acid and a strong base and they're monoprotic, the equivalence point is going to be at 7. So that's for a strong acid and a strong base. If I had a weak acid and a strong base, I'm going to get a curve that has an equivalence point that is higher than 7. And if instead I had a strong acid and a weak base, the equivalence point will be less than 7. So pH of 7 is sort of, we'll think of that for a strong acid and a strong base. If it's a strong acid and a weak base, the pH of the equivalence point will be less than 7, but think, strong acid. And if it's a weak acid and a strong base, the equivalence point will be greater than 7, think stronger base. So it's going to be a more basic pH for the equivalence point. And you'll notice on here, we don't have listed weak acid and weak base. They don't make for good titrations. So there should never be a question that references that. So this is good because you should be able to get a general idea of what the equivalence point should be, depending on whether it's a strong acid or a strong base, or and whether it's a weak acid and a weak base.